Hello everyone, welcome to my YouTube channel Learn Life Science. So in this particular video we will be learning about the quantitative determination of RNA, that is ribonucleic acid. Basically the ribonucleic acid quantitative estimation can be done by two methods. One, by using a kind of reagent, that is Orcinol reagent, and therefore it is also known as Orcinol method for quantitative determination of RNA. Second method is by taking directly the UV absorbance of the sample, okay, by which you can detect the concentration of RNA using a particular formula. Okay, so we will be first discussing the Orcinol method. Okay, first we will be discussing about Orcinol method, and in this case, generally, Orcinol is a type of reagent. It is a reagent which reacts with hydrolyzed product of ribose sugars, okay, or hydrolyzed product of pentose sugars, right? For example, ribose. Okay, so when a pentose sugar, for example, ribose, is heated with the concentrated hydrochloric acid, it forms an intermediate product named as furfural. Okay, so in this case you can see the ribose in acidic condition, and when it is heated in acidic condition there is dehydration of the ribose sugar. Okay, you can see the dehydration causes the ribose to get transformed into furfural. Okay, now this furfural intermediate can react with Orcinol reagent. It can form complex with Orcinol reagent, and this reaction is increased or enhanced by using a catalyst, that is ferric chloride. Okay, so in the presence of ferric chloride, the reaction is going to be enhanced, or the rate of reaction is going to be increased. When Orcinol and furfural, both of them will form a complex, and they will give you a blue-green colored condensation product. Right? So this was a method generally utilized for the analysis of ribose sugar in the sample, or pentose sugar ribose in the sample. Right? Okay, so basically ribose, it is a aldopentose. Okay, ribose is a aldopentose sugar. Aldopentose means it is a 5-carbon sugar in which the functional group present is aldehyde, that is in the form of CHO, right? So basically the aldopentoses sugars like ribose, arabinose, lysose as well as xylose can react with orcinol when they are treated with hydrochloric acid, or when they are treated in acidic condition, okay, and high temperature in acidic condition. Okay, talking about other sugars, for example, if we talk about ketopentoses or ketohexoses, those having keto group as a functional group, for example, ribulose and xylulose. These kind of sugars having keto group generally react with resorcinol reagent, right? In my previous video, I have mentioned about Selivanov test, in which I have discussed that which sugar generally reacts with different reagent or resorcinol reagent specifically. Okay, so keeping this idea in mind, means now we know that ribose is a sugar which in acidic condition, when heated in acidic condition undergoes dehydration, and the intermediate product condenses with the reagent to form a blue-green colored product. Keeping this idea in mind, now we can use the same method for the quantitative estimation of RNA. How? Because we know that RNA, that is ribonucleic acid, consists of the sugar in the form of ribose, right? Ribose is the sugar which is present in the RNA structure. So on the basis of this, you can quantify the amount of ribose sugar which is present in your sample containing RNA. Okay. So now we will be studying the principle of quantitative estimation of RNA by Orcinol method. In this case, you can see among the various colorimetric methods for RNA estimation, the Orcinol reaction is the most sensitive method for purine-bound ribose quantification. This method again is going to give you the amount of RNA only which is associated with the purine bases. Okay, why? I'm also going to mention that. Okay, in this method, RNA is first depurinated in concentrated HCl, and the resulting ribose phosphates are then dephosphorylated, and further it is dehydrated to produce furfural. Okay, so for example, this is the structure of nucleotide. It is a RNA nucleotide. Okay, so first just look at the structure. In this structure you can see here the base, nitrogenous base, is linked with the ribose sugar. The bond between the base and the sugar is glycosidic bond. Glycosidic bond, right? Basically the bond between sugar and purine. Okay, if I write here, the bond between the sugar and purine when compared with the bond between sugar and pyrimidine. The most stable bond between sugar and base is here. Okay, in case of sugar and pyrimidine, the bonding is more stable and strong. Okay, in case when the sugar is linked with the pyrimidine base, that is thymine as well as cytosine. When it comes to sugar and purine bond, when you will heat the solution in the presence of acid, the bond between sugar and purine can easily break down. Okay, so therefore in this method they have mentioned that the RNA which is the sugars which is linked with the purine base are going to get depurinated, and not those sugar which are linked with the pyrimidine. Okay, so this is the first point. Moving to the second point, what generally happens in the presence of acid and in the boiling condition at high temperature, there will be depurination, there will be removal of the purine base, and the second one will be dephosphorylation. Dephosphorylation means removal of the phosphate groups as well. Okay, so here is the phosphate group. It is also going to get removed out from 3' as well as the 5'. Okay, now what remains is only the sugar, right? Now this sugar undergoes dehydration. Okay, now this sugar is going to undergo dehydration, and on dehydration there will be formation of an intermediate which is termed as furfural. Okay, so this furfural then reacts with orcinol in the presence of ferric ions to yield a colored condensation product which together possesses an absorption maxima at 660 to 665 nm. Okay, and therefore we can easily measure the absorbance of the end product at 665 nm. Remember, the color intensity is directly proportional to the concentration of ribose present in the sample of RNA. Okay, so I hope you are clear with the concept behind the reaction. Now we will be moving towards the requirement that you are going to require to perform the protocol. So first thing, we are going to require an RNA standard, means you are going to prepare a standard graph, right? And in this case you will require a standard RNA which is available in the laboratory. So for example, we are taking the concentration of standard as 100 microgram per ml. If you convert it in milligram for 100 ml, it will give you 10 milligram in 100 ml solution, right? So you will have to prepare the RNA in a buffered saline. Okay, buffered saline, you can see here I have mentioned the saline composition. Okay, so you will have to separately prepare the saline, and you will also have to prepare the saline for the RNA standard preparation. The second thing will be the requirement of Orcinol reagent. Okay, so for sodium saline citrate preparation, you will require 0.15 molar of NaCl, and you will require 0.015 molar of sodium citrate. Okay, you will have to prepare both the solution in 100 ml. Okay, that will give you your saline sodium citrate solution. Okay, now for Orcinol reagent preparation, how you are going to prepare Orcinol? Orcinol is commercially available in the form of powder. Okay, using that you will have to further add ferric chloride as well as acid. Okay, because we have to maintain every condition, right? So first thing is you are going to prepare 6% Orcinol solution. For preparing 6% of Orcinol solution, you are going to dissolve 6 gram of Orcinol in 100 ml of ethanol. Orcinol is going to dissolve in ethanol. Okay, and you will have to store it at room temperature in a amber colored bottle. Okay, you will have to prevent it from photo oxidation, and therefore we are storing it in dark colored bottle. Second thing is how you are going to prepare the reagent. The reagent is going to consist of three components: orcinol, ferric chloride, as well as the HCl. Okay, so in this case you will have to dissolve 0.1 gram of ferric chloride in 100 ml of concentrated HCl. After that, you will have to take 3.5 ml of the 6% orcinol in ethanol to it. Means you will have to take 3.5 ml of orcinol which you have already prepared, and you will have to add to this solution. Okay, and remember, this reagent should always be prepared freshly prior to use. Okay, so this is about the requirement as well as the reagent. Moving on, next we will be discussing the protocol. So this is the performing set. In this case we have taken 6 test tubes, okay, and 1 blank. Okay, so we have taken 5 test tubes which is considered for the standard graph, and 1 test tube which is containing your unknown sample. Okay, so the RNA stock solution which you have prepared with a concentration of 100 microgram per ml, right? This is your concentration of RNA stock. So we will be taking 0.2 ml, 0.4, 0.6, 0.8 and 1.0 ml of the standard RNA from the stock solution in the test tube. Okay. Also, we are knowing what is the concentration of RNA stock. Okay, so therefore we can calculate if 100 microgram is for 1 ml, that means for 1 ml the concentration mentioned is 100, right? So similarly for 0.8 ml it is going to be 80, for 0.6 ml it is going to be 60, for 0.4 it is going to be 40, for 0.2 it is going to be 20. If you want to take 10 test tubes, for example 0.1, 0.2, 0.3, 0.4, 0.5, 6, 7 and 9, 10, so you can take like that. Okay, so for that the concentration will be like this. For example, you have taken 0.1, the concentration of RNA will be 10. For 0.2 the concentration will be 20, and when you will reach 1.0 the concentration will be 100. Okay, because we have taken the standard concentration. Okay, so you have prepared the standard tubes. The sixth tube which is containing your unknown sample, in that you will have to take 3 ml of your unknown sample. Okay, in this case we are also going to make up the volume up to 3 ml using distilled water, and for this purpose you will have to add 3 ml of distilled water in the blank test tube, then 2.8, 2.6, 2.4, 2.2, 2.0. Okay, then finally you will have to add the orcinol reagent 3 ml in each test tube. Okay, in each test tube, in unknown test tube, in the blank test tube, as well as the standard test tubes. Okay, then we are going to incubate it. The incubation is going to take place in a boiling water bath because we are requiring high temperature for the reaction to occur, right? So you will have to keep the test tubes in boiling water bath for 20 minutes and then cool down to room temperature, or you can just cool down in under running tap water. Okay, and finally we are going to take the absorbance at 665 nm. Okay, I have taken random readings. We are going to use these readings for the preparation of standard graph. Okay, so okay, so this is your standard graph. Okay, on x-axis you can see, on x-axis it is mentioned RNA concentration which is in microgram per ml, and on y-axis absorbance is present which is taken at 665 nm. Okay, on plotting the graph you can see I have plotted at 20 concentration of microgram per ml, the point is 0.028. Then here the point is 0.056, 0.0842, 0.111 and 0.14. Okay, in this manner I have got a straight line. This straight line is from these 5 readings. Okay, so the upper standard tube 5 reading will give you the straight line. Okay, for example, your unknown test tube. For example, your unknown test tube gave you reading of about 0.062. Okay, for example, it gave you reading about 0.6062. So how you are going to calculate the accurate concentration of this sample, right? So for this, we are first going to calculate the slope. For calculation of slope, the formula is y2 minus y1 divided by x2 minus x1. For example, I am taking y2 as 0.111 and y1 as 0.0842. Then x2 will be 80, x1 will be 60. In one of my video, I have already mentioned, already prepared a video on how to calculate the slope. Okay, so you can refer that video also. So taking the values, we will write here 0.111 minus 0.0842, right? y2 minus y1. Similarly, x2 minus x1 is 80 minus 60. Okay, so I'm writing here. It will give you a value of around 0.0268 divided by 20. The answer will be 0.00134. This value is your slope. Okay, this is your slope. To calculate the unknown concentration, you can use the formula OD by slope. Okay, your absorbance, your OT is 0.062. Okay, I have mentioned here 0.062 divided by the slope, that is 0.00134. The answer will be 42.26. This is the accurate concentration of the RNA which is present in your sample. Again, this is the concentration of only that amount of RNA which is having the purine base. Okay, so you can multiply it by 2 to get the total concentration of the RNA which is present in your sample. Okay, so this is how we generally calculate the concentration of RNA. The second method to calculate the concentration, I have already mentioned that you can directly plot it on the y-axis. For example, it is 0.062. So if I plot it right here on y-axis, I will take a straight line and I will take it right here. Okay, so for example, this is 50 and it will give you around 40. Your accurate measure can be get around 42. Okay, so this is another method to calculate the unknown concentration. Okay, so this was all about the Orcinol method for quantitative estimation. The second method, that is the quantitative estimation by taking the UV absorbance of the sample. Okay, so we will be discussing that point also in short. So RNA measurement can also be conducted by measuring the ultraviolet absorbance at 260 nm and also at 280 nm. The calculation of the RNA concentration is based on the absorbance at 260 nm. Furthermore, RNA purity is judged as the 260 nm by 280 nm ratio, and the low ratio indicates contamination by protein. What is mentioned in the statement is, you can see in this graph. In the graph, you can see the highest peak is observed at 260 nm. 260 nm, if a peak is observed at 260 nm, it indicates that RNA or even DNA is present in your sample. Okay, the absorbance is undertaken by the nitrogenous bases which is present in the nucleic acid. It gives us maximum absorbance at this point. The second point, talking about 280 nanometer. If you observe a peak at 280 nanometer, for example, if I prepare another graph like this, we'll change the color. If I got another graph like this, okay, for example, I have taken this. So you can see two peaks. You will see two different peaks. One peak at this point and one highest peak at this point. Means at 220 nm. So this 280 nm peak, the peak at 280 nm indicates the presence of proteins. Okay, and mostly the aromatic amino acids. The second peak which you are observing at 220 nm is due to the absorbance by peptide bond. Okay, this is due to the absorbance by the peptide bonds which is present in the proteins. Okay, So whenever you are taking RNA UV absorbance, if you are observing a single peak at 260 nm, it means that your RNA is free from any kind of contamination by protein. Okay. Also, we take the ratio of both. Ratio of absorbance at both 260 nm and 280 nm. Okay, if the value is 1.8, it indicates that your sample is purely containing the RNA sample. Okay, and if the ratio is low, if the value is less than 1.8, it means that the sample is contaminated by proteins. Okay, so therefore here the ratio at 260 nm is divided by the 280 nm where proteins gives us maximum absorbance. Okay, so this is the idea regarding UV absorbance. Also, there is one specific formula for calculation of RNA concentration, and that is, okay, the standard mentioned here is absorbance at 260 nm of 1. If you get reading as 1 at by taking absorbance at 260 nm, it means that the sample concentration is equivalent to 40 microgram per ml. Okay, this is a standard value which is mentioned, which is given for pure single standard RNA. Okay, so by taking this value in mind, we can calculate the concentration of RNA. Okay, so the concentration of RNA in microgram per ml can be given as absorbance, which you have taken at 260 nm. Okay, then you will have to multiply it by the dilution factor. If you have diluted your sample, you will have to consider that too. Dilution factor is nothing but the initial volume of your sample divided by the final volume of your sample. Okay, then you will have to multiply it by the standard value, which is 40 microgram per ml. Okay, the value you will get here will be the concentration of your sample. Okay, this is going to be the total concentration of the RNA which is present in the sample. Okay, so I hope you are clear with both the concept, with the Orcinol method as well as by the UV absorbance method. Okay. Okay, and still if you are having something to share regarding the protocol, you can still mention in the comment box. Don't forget to like, share and subscribe. Thank you so much for watching.